Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International A-Level Biology Unit 1 for January 2023. Let us begin with the first question. Question 1. Water and carbohydrates are molecules found in all living organisms. Draw a diagram of a water molecule to show its dipole nature. So of course, water contains one oxygen and two hydrogens, and each bond is going to be polar. So there is a partial positive here, a partial negative there, and a partial positive here. And the bond angle is 104.5. Of course, this wasn't required, but I just placed it there. In part B, the table gives some statements about carbohydrates. For each statement, put one cross in the appropriate box in each row to match the statements to the correct carbohydrates. So here they say, have the general formula carbon N, hydrogen 2, and oxygen N. These are monosaccharides only, so this is my answer. Have a glycosidic bond. This is for polysaccharides only, we can see that. And then have a little effect on water potential. That is going to be for polysaccharides only. So this brings us to the end of question 1. Let's continue to question 2. Question 2. Elastin and collagen are proteins found in connective tissue. They want you to describe the structure of collagen. Collagen is a fibrous protein, and of course it's made up of 3 polypeptide chains. The polypeptides are 3 strands that are used to form a helix. So the chains are held together by hydrogen bonds, and polypeptide chains are mainly made of repeating sequences of glycine and other amino acids like proline as well as hydroxyproline. In the next part, they say proteases are enzymes that break down proteins. The graph shows the effect of protease solution on a sample of elastin. So on the vertical axis, we see the concentration of breakdown product in nanomole per decimeter cubed. And on the horizontal axis, we can see time. So next they say, calculate the rate of reaction at 30 minutes. Draw a tangent to help with your calculation. So all I needed to do is to come at 30 minutes, which is that, and draw a tangent at that point, and then find the gradient of that tangent. As you can see, the points are used, where that point which is 2140, and we have a 50 to 10. So I'm going to say 210 minus 140 divided by 50 minus 20. So 210 minus 140 divided by 50 minus 20, it gave me 2.33. The acceptable answer is between 2 to 3.158. So this was my answer. Next they say, explain how a protease breaks down elastin. Since the protease is going to be the enzyme and this is going to be the substrate, for the breakdown to occur, it has to bind into the active sites. So I said, elastin binds into the active sites of protease, forming an enzyme substrate complex, and the activation energy for the reaction is going to be lowered. The peptide bond is then broken through a hydrolysis reaction. So this brings us to the end of question 2. Let's continue to question 3. Question 3. Cystic fibrosis results from mutations in the CFTR gene. Mutations may occur at a number of positions within the gene, resulting in a range of clinical symptoms and severity of cystic fibrosis. The effect of combining 3 different drugs to treat people with cystic fibrosis was investigated. People with cystic fibrosis were divided into 2 groups. One group was given the 3-drug combination, and the other group was given a control treatment. The volume of air each person could forcefully blow out in 1 second was measured. The graph shows the results of this investigation. The group given the 3-drug combination had an increase in the mean volume of air blown out, but for the other 2 there was some decrease. So they say, explain why the change in mean FEV was used in this investigation. Remember, FEV is about volume of air each person could forcefully blow out in 1 second. Now, because this is studying cystic fibrosis and we know it affects or damages lungs, it affects lungs and causes them to be blocked, so the surface area for gaseous exchange is going to be decreased. This method should have been used in order to study the efficiency of how the medicine is trying to clear out these problems within the lungs. So I said, cystic fibrosis causes the production of thick sticky mucus. The mucus builds up in the lungs, blocking the airways, so less volume of air goes in or out of the lungs. When the treatment is given, there is improvement in FEV. Since mean results were used, validity of the data can be established. Moving on. Explain why the results of this investigation do not show that the treatment is more effective than using each drug on its own. Based on the graph, you can see there were no results given for each individual drug, so we cannot make a conclusion about that. So I said, the investigation did not give the results for each drug, so there is no way to compare to find out which one is more effective. In part C, suggests why a combination of drugs could be more effective in treating people with cystic fibrosis than each drug used on its own. As we know, cystic fibrosis affects different organs or different organ systems. It affects the gaseous exchange system, including the lungs, differently. It affects the gastrointestinal system differently, as well as the reproductive system differently. So I said, cystic fibrosis is caused by different mutations, and these mutations affect the formed CFTR protein differently. This affects organs differently, so different drug combinations should be used for treatment because each could treat a different symptom. So this brings us to the end of question 3. Let's continue to question 4. Question 4. The diagram shows the internal structure of a mammalian heart. How many of the following are correct for blood vessel P? Blood vessel P is the vena cava. So they say carries blood to the heart, correct? Has valves along its length. It's correct because that is a vein. It has an endothelial lining. That is also correct. So the answer should be D. 3 are correct. And next they say, why is blood vessel Q classified as an artery? Let us go back to blood vessel Q. That is the pulmonary artery. It is classified because it carries blood away from the heart. You could say that. So the answer here is going to be a C. Moving on. Here they ask, which blood vessel does the coronary artery branch from? If we go back, the coronary artery is going to branch from the aorta, which is going to be this blood vessel R. So my answer here was C, which is R. The next they say, when in the cardiac cycle is valve T open? This is valve T, the bicuspid valve. It is going to be open during atriosystole, meaning when the artery are contracting so that blood goes into the ventricles, as well as in cardiac diastole. So the answer here should be an A. Moving on. Here they say, the effect of atropine and propranolol on heart rate was investigated. So one group of people, group 1, were given propranolol each day for 4 days, followed by atropine for 4 days. A second group of people, group 2, were given atropine each day for 4 days, followed by propranolol for 4 days. The graph shows the results of this investigation. So we can see there are two groups. This drug was given here to group 2, and then this drug given here and given there in different groups. So we can see basically the drug given here causes an increase in the mean heart rate, and this drug here causes a decrease in mean heart rate. Although at the end there was no significant difference because the result is almost the same no matter which drug was given first. They say calculate the change in mean heart rate for group 2 after 8 days. Going back here, for group 2, which is this one here, initially it was about 62 beats per minute. And at the end, which is this one here, at the end is going to be 94 beats per minute. So the overall change in heart rate is going to be 94 minus 62, which gave me 32 beats per minute. So that is the change. And next they say calculate the mean length of the cardiac cycle for group 2 on day 4. So if we go back on day 4, we can see the mean heart rate is about 111 beats per minute. So when I came here, we have 11 beats per minute or 111 beats in 1 minute. We know 1 minute is 60 seconds. So this is going to be the same 111 beats in 60 seconds. And in 1 beat, it's going to be 60 divided by 111, which is 0.54 seconds. So we can say the length of the cardiac cycle is going to be 0.54 seconds. And down here they say comment on the conclusions that can be drawn from this investigation. Now, like I already said, of course, we see atropine increases the heart rate. We can see for both groups. And then we see propranolol decreases the heart rate. But overall, we see there is no significant difference. It doesn't matter which drug was given first or which one was given later. The overall results did not have a significant difference because we see there is overlap in error bars towards the end. So I said, propranolol decreases the heart rate while atropine increases the heart rate. This increase and decrease occurs despite the order in which the drugs are given. At the end, the overall change in heart rate is the same for both combinations. And again here they said comment, so I have to talk about the validity. So I wrote, since means were calculated and error bars are shown, the results are valid. However, for the last 2 days, error bars overlap, so no validity. Also, the error bars for atropine are wider until day 4, so validity may not be guaranteed. So this brings us to the end of question 4. Let's continue to question 5. Question 5. The portfolio diet is a diet that has been shown to lower low-density lipoproteins and other risk factors of cardiovascular disease. Which row in the table describes the cholesterol content and antioxidant content in this diet? Because it lowers low-density lipoproteins, it means it's going to have a lower concentration of cholesterol. And because it lowers risk factors for cardiovascular disease, it's going to have a high concentration of antioxidants, so the answer is going to be a C. In part 2, name 2 other dietary risk factors that could be changed in this diet. So for this diet, we could change the saturated fat, we could change the salt intake, the quantity of fiber, and so on. So any 2 of these would earn you 1 mark. Moving on. In part B, the effects of this diet on the development of CVD in women was investigated. The diagram shows 123,330 women were selected from an initial group of 161,808 women. This is the initial group, and then these women were removed from the group, and we remained with these ones. More were removed, and then finally they had the final group. They say some of the women were removed from the initial group because there was information missing about their lifestyle. Suggest why women with missing lifestyle information were removed from the initial group. We know that lifestyle factors have an effect on CVD, so including the women whose lifestyle factors were not included would make the investigation invalid. Moving on. Here they say some of the women were removed from the initial group because they gave unrealistic estimates of their daily energy intake. Give 2 reasons why some women would have given unrealistic estimates of their daily energy intake. It could be that they were embarrassed because they consumed a lot of calorie-containing foods, or they never recorded or they did not remember their daily intake. So I said they did not record their daily dietary intake. Or the women could have consumed a lot of high energy or cholesterol-containing foods, so they were embarrassed. Next, they say the women removed from the intermediary group had signs of CVD. Explain why these women were removed from this investigation. Because they already had signs of CVD, it means the investigation will not be valid. So I said the investigation was studying the effect of diet on CVD. If some women already had signs of CVD, they were developing the disease, meaning the diet they are trying to investigate is not what is causing them to have the CVD. So then the investigation would not be valid. Down here they say, calculate the percentage of all women removed from this investigation. So if we go back here, these women were removed as well as those, so I added them together. So that plus that gave me this. That divided by the total times 100 gave me 23.78, which is about, you could say 24% or 23.8%. That is a percentage. So here they say, explain why a diet that is low in saturated fat is likely to reduce the risk of CVD. If it's low on saturated fat, it means it contains less cholesterol, so there is lower chance of formation of atheromas due to cholesterol being deposited around blood vessels. So I said, because a diet in low saturated fats results in less cholesterol in blood, therefore less plaque or atheromas are formed, so there is less blockage in the coronary artery, so the heart muscle will get sufficient oxygen and nutrients for aerobic respiration in the cardiac muscle cells. So this brings us to the end of question 5. Let's continue to question 6. Question 6. The fluid mosaic model of membrane structure was developed to explain the structure and properties of cell membranes. What model of membrane structure was developed by Devson and Danieli? The diagram shows the Devson-Danieli model. They say which describes a phospholipid. A phospholipid has a hydrophilic phosphate head and hydrophobic fatty acid tails. You can see there are 2 tails on one head. So moving on. Here they say, compare and contrast the structure of the model shown in the diagram with that of the fluid mosaic model of the membrane structure. So if we go back here, we can see, compared to the fluid mosaic model and this model, we can see both are made up of phospholipids, both have bilayers. However, in this model, we can see there are proteins all over. They're not intrinsic proteins, but they're surface proteins. We can see they're on the surface. So I said similarities, both have phospholipid bilayers and both have proteins. The differences are, in the fluid mosaic model, the proteins are embedded in the layer, meaning intrinsic proteins, while in the Devson-Danieli model, the proteins are only found on the surface, meaning they are extrinsic. The fluid mosaic model has cholesterol, while the Devson-Danieli model does not. The fluid mosaic model has glycolipids, while the Devson-Danieli model does not. Down here they say, the cell membrane is important to the cell, and if it gets damaged, the cell could die. Suggest how damage to the cell membrane could result in cell death. This is easy. The cell membrane controls what enters or leaves the cell. So if there is damage, then there could be uncontrolled movement of substances, and this could cause problems within the cell. So I said damage could result in uncontrolled movement of substances like water, salts in or out. The concentration gradients will no longer be maintained, and cellular processes like respiration could stop, leading to cell death. And if the cell lost so much water, the cytoplasm can dry and the cell could die. And if the cell took in so much water, it could burst. Moving on. Cells can repair damage to the cell membrane. This involves fusion of parts of the cell membrane around the edge of the damage. Explain why parts of the membrane are able to fuse together to repair the damage. This is mainly because the phospholipids can freely move and they will interact with each other, meaning the hydrophilic heads will interact with each other and the hydrophobic tails will interact with each other, causing the fusion around the damage. So I said the phospholipids can freely move within the membrane, and the fatty acid tails interact with each other. The hydrophilic phosphates also interact with each other to fuse together. Down here they say, once the membrane has fused together, other processes are needed to complete the repair of the membrane. Suggest what else the cell needs to do to complete this repair. The cell needs to add more proteins. The cell needs to produce more ATP to carry out the processes of like additional more proteins and so on. So I said more respiration for ATP required for the repair, additional more intrinsic proteins, and additional more glycoproteins or phospholipids, as well as glycolipids. So this brings us to the end of question 6. Let's continue to question 7. Question 7. The photograph shows a yak. So this is the yak here. Yaks are cattle that live at high altitudes such as in the Himalayas. They are well adapted for the low temperatures and low levels of oxygen in the air. These adaptations include a thick layer of fat below the skin, large lungs and a large heart, many small red blood cells, presence of fetal hemoglobin throughout their adult life. In part A, triglycerides are fats. Describe the structure of a triglyceride. Triglycerides have 3 fatty acids and 1 glycerol, and these are joined together by condensation reaction to form an ester bond. So you could include by condensation reactions. So moving on. Here they say, explain why large lungs and a large heart would help a yak to survive at high altitudes. Large lungs enable you to take in large volumes of air. Even if the partial pressure of oxygen is lower, it means when they take in more volume of air, they're going to have more oxygen intake. Also, a large heart allows them to pump greater volumes of blood for a small period of time. So I said, the large lungs allow them to take in large volumes of air into the alveoli, and they have more alveoli, so the surface area for gaseous exchange is going to be high, or you would say large. The large heart allows for more blood to be pumped per minute, so organs receive sufficient oxygen for aerobic respiration. Down here, the table shows some characteristics of blood of yaks and the blood of humans. So we can see we're going to compare yaks and humans. The concentration of red blood cells, we can see 1 times 10 power 13. This is higher compared to that in humans. The volume of one red blood cell, we can see it's smaller in comparison to the humans. Then the concentration of hemoglobin, it's quite high compared to the humans. So in conclusion, we can see they have higher concentration of red blood cells. Their red blood cells contain less volume and then they have a higher concentration of hemoglobin. So here they say the unit given for volume of red blood cells is femtoliter, where 1 femtoliter is 1 times 10 power negative 15 decimeters cubed. They say give the volume of red blood cells of a yak in centimeters cubed. We know 1 femtoliter is 1.0 times 10 power negative 15 decimeters cubed, but 1 decimeters cubed is equal to 1,000 centimeters cubed. It means to convert this into centimeters cubed, I can multiply that, and I got 1.0 times 10 power negative 12 centimeters cubed. So 43 femtoliters are going to be 43 times that, which gave me 4.3 times 10 power negative 11, and that is my answer here. Moving on. Here they say the diagram gives some information about two spheres. There is sphere A and sphere B. So we can see sphere A has a diameter of 8 millimeters, and this one has a diameter of 16 millimeters. Sphere A has a surface area to volume ratio of 1 to 1.3, and sphere B has a surface area of 768. They say calculate the surface area to volume ratio of sphere B, and they want us to use this formula. So at first, I found the volume of sphere B, which is, using this formula, is 4 over 3 pi r cubed, where my radius is going to be 8 because the diameter is 16. So substituting, I got 2048 millimeters cubed. Now this is my volume, and I have the surface area here. So surface area to volume ratio is going to be 768 divided by that, which is the same as 1 to 2.7, or you could round off to 1 to 3. So my answer here is 1 to 2.7. Moving on. Here they say, discuss why the blood of yaks enables these animals to live at high altitude. They say use all the information in this question to support your answer. So I'm going to go back once just to show you. So we can see from the beginning, we had this information here. Remember, yaks have thick layers of fat below their skin. They have large lungs and large hearts. They have many red blood cells. There is presence of fetal hemoglobin throughout their adult life. When we come to the table, we can see they have a higher concentration of red blood cells. They have smaller volume of red blood cells, and then they have a higher concentration of hemoglobin. So I'm going to use some of this information in my answer. From the table, they have a higher concentration of red blood cells, and this allows for more oxygen to be transported to the organs for aerobic respiration. Also from the table, they have small volume of red blood cells. This provides a high or large surface area to volume ratio for the diffusion of oxygen. However, not so much oxygen can be packed per cell. This can, however, be overcome due to the large numbers of red blood cells. From the table, they have a high concentration of hemoglobin, so more oxygen can be transported to the tissues for aerobic respiration. And also we saw that their blood contains fetal hemoglobin until adulthood. Since fetal hemoglobin has a higher affinity for oxygen, even at low partial pressures, they therefore carry more oxygen. So this brings us to the end of question 7. Let's continue to question 8. Question 8. Red blood cells have a number of different groups of proteins on the cell surface membrane. The inheritance of some of these groups of proteins is an example of codominance. The pedigree diagram shows the blood type of individuals in part of one family. There is individual 1 and individual 2 who are both homozygous for blood type. So we have the key. They're homozygous. They've told us here they're both homozygous. So it's going to be capital M, capital M. And the female blood group N, capital N, capital N. So it means their child is going to have codominance because they're going to be capital M, capital N. So they say, explain why the inheritance of blood type shown in this diagram is an example of codominance. This is quite easy. Because neither alleles are dominant, or we could say both alleles are expressed. So when we use individual 3 as an example, both alleles from individual 1 and 2 are present, but both are expressed in the phenotype. So moving on. Here they say, determine the ratio of the possible blood type phenotypes of the children of individual 3 and 4. You must draw a genetic diagram. The genotype of individual 3 and 4 is going to be capital M, capital N. So their gametes are going to be either capital M or capital N. So in this case, for one individual, capital M, capital N, and capital M, capital N, the genotype of the offspring is going to be capital M, capital M, capital M, capital N, capital M, capital N, and capital N, capital N. So we can say the phenotypic ratio is going to be 1M. Remember, phenotype is the outside appearance. There is going to be 1 capital M, 1 capital N, and 2 capital M, N. So moving on. The gene that determines this blood type codes for proteins present on the cell membranes of red blood cells. The diagram shows red blood cells from 2 members of the family shown in the pedigree diagram. So we have protein M as well as protein N. This is individual 1 and that is individual 2. Explain how the alleles present on individual 3 results in the proteins found on the surface of his red blood cells. So the alleles are going to be transcribed into messenger RNA. This messenger RNA is going to be translated into a protein, and then the protein is going to be incorporated onto the surface of the cell. So I said individual 3 has 1 capital M allele and 1 capital N allele. Both are going to be transcribed to messenger RNA. Then translation occurs to produce the surface protein. Then these surface proteins are incorporated onto the surface of individual 3's red blood cells. Moving on. Here they say, discuss the possible effects that a mutation in one of the genes coding for this blood type could have on the phenotype of individual 3. So mutations can be substitution, they can be deletion, or they can be insertion. So I said, if a substitution mutation occurs, a different amino acid may be placed in the wrong position in the polypeptide chain, and this can affect the shape and function of the protein. If a stop codon is formed due to the substitution, then the formed protein will be shorter and lacking the required amino acids to form the functional 3D protein. If the substitution occurs in the third base of the codon, the amino acid sequence may be unchanged since the genetic code is degenerate, so the protein will be the same. If insertion mutation occurs, a base is added, the DNA frame shifts by 1 position forward, and if a deletion mutation occurs, a base is removed, and this will cause the shifting of the DNA by 1 position backwards. So in both these shifts, the formed polypeptide chain is different, and the protein will be different as well. If the formed phenotype becomes the gamete, then the offsprings will have the allele. So this brings us to the end of question 8 as well as to the end of this paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.